My name is Andrew Waterhouse, Director of the Robert Mondavi Institute and a Professor of Enology. Welcome to this evening's forum program. As a public university, we pursue a mission of excellence in education and research for the benefit of California and the world. Part of our mandate is to disseminate knowledge and foster dialogue with other experts and the public through events such as this one. At the Aroma, we feel it is important to enhance public understanding of wine and food science by sponsoring the exchange of ideas. Along those lines, please submit your questions for our speakers using the Q&A tool throughout the program. We will try to answer as many questions as possible at the end of our program today. Forum presents an array of research and scientific advancements in the disciplines of wine, brewing, and food science to demonstrate the importance of UC Davis research to the California economy. Today's program is of particular interest because Mr. Mondavi was a great advocate of the latest science. And today, the most exciting and powerful scientific advances are occurring in the field of genetics. Before we move on, I wanted to mention that we have a list of events coming up that I will describe at the end of our program. Also, this program is being recorded and will be posted on the RMI website about midweek next week. So your friends who could not attend will be able to watch the recorded version. Today we will hear from Dario Cantù, Professor and Martini Endowed Chair in Viticulture. Professor Cantù earned his PhD in plant biology at UC Davis studying the botrytis tomato fruit interaction and subsequently worked with Professor Dubkovsky on wheat genomics and the striped rust wheat interaction. The Department of Viticulture and Enology hired Dr. Cantù in order to bring these skills, technologies, and new capabilities to the study of grapes. He's one of our top researchers and has been recognized as a Chancellor's Fellow here at UC Davis and also serves as the Director of the UC Davis Chile Life Sciences Innovation Center. His research focuses on plant and microbial genomics with a focus on wild and cultivated grapevines and grape pathogens. I will now hand the microphone over to Dr. Cantù, but suffices to say that he has been instrumental in organizing the interdisciplinary research teams that have mapped both the Cabernet Sauvignon and Chardonnay genomes. Dario.
Thank you, Andy, for the kind introduction and for giving me the opportunity to present here part of my research program. And even if it's not the ideal format, I'm always impressed by the number of attendees and the fact that they come from multiple countries, and it's really amazing how many people you can reach with this type of interactions. Let me share my screen so I can start. Okay, so my presentation today is going to focus on what we've been doing in grape genomics. And before I start talking about actually grape genomics, I would like, let me share the full screen here. I would like to introduce genomes. And so we are all on the same page in what I'm talking about. The genome in very simple terms is the complete genetic information of an organism. This genetic information is stored in the DNA of that particular organism. So when we talk about mapping genomes, sequencing genome, we are basically looking at the chemical composition of the DNA to gather all the genetic information that particular organism possesses. So if you think about grapes, the genetic information, the genome of grapes is organized in 19 chromosomes. And every time we sequence this DNA information, we sequence about 500 million letters, okay? These 500 million letters encode for a large number of genes. These genes are the very important information that provide the details to produce the proteins that will be responsible for the development of the grapevine, their photosynthetic capacity, as well as the development of the fruit and all the things we enjoy about grapes. Just to put into context these numbers and making some comparison with what you know better, for example, our own genome, the grapevine genome is about 1/6 of the information, the genetic information we have. However, if you look at the number of genes, we are basically encoding a very similar number. And if you compare now to the bread yeast, the bread yeast is about 40, 50 times smaller than the grapevine. But does it really matter the size? And I like, to talk about COVID at this point because COVID has been has had quite an impact on our lives, and its genome is very tiny and it's very simple. So in genomics, size matters to a certain extent. However, when we think about grapevines, we're dealing with an average genome size with a good number of gene encoding proteins, similar to what our own genome does. So oftentimes we think about the genome as one thing, the human genome, the grape genome. However, there is no genome equal to another genome. Every genome is different from another. So even if you compare grapevine clones, you have genetic differences that accumulate over time as mutation. When you compare varieties, you will find more genetic differences, and you find even more genetic differences when you compare different species. These genetic differences, so differences in the DNA composition that result in differences in genetic information, can be very subtle, like I'm showing here, differences in letters, or can be more substantial with big changes in chromosome structure, which we call structural variant, which are becoming very important also in human genomics as relevant disease-causing mutations. Why do we care about this? Why do we care about gathering genetic information from a grapevine, sequencing their genomes, and comparing them? Well, a basic question we can ask, well, what are the genetic differences between two varieties? As you go to the store and pick a Cabernet Sauvignon, you're choosing a genome. You're choosing the genetic information that made that Cabernet Sauvignon. Obviously, the environment plays a role as well, but you're not choosing a Chardonnay, which means that you're not choosing the Chardonnay genome. So what are the differences now between those two genetic makeups that make you choose one or the other? So I give you just one example. It's not from my lab, but it's a clear example of how a small change in the genome can make a big change in how we appreciate the grapes and the resulting wine. What I'm showing here is Pinot Noir and Pinot Blanc. They have different skin color. Well, this different skin color is due to the deletion of a single gene in the genome. Okay, I talked about 30,000 genes, 19 chromosomes. Well, a small change in one specific region of the genome causes a big change in how the grapes look like and the wines we can make from them. So over the years, and as Andy mentioned, I started in 2012, and early on in my research, I started gathering collaborations and the funding to begin developing tools to study the grape genomes. And one key aspect to this was to be able to reduce the cost so we could start appreciating the genomic diversity across the grapevine germplasm. When I started in 2012, there was already a genome reference, a single genome reference. Having a single genome reference was super useful, but did not represent the genomic diversity in the germplasm, so didn't allow us to actually appreciate the genetic differences responsible for the phenotypic diversity we observe. So these are just some of the titles of the articles we published over the years. And obviously, once we started having these tools available, we could explore further very fundamental questions from really understanding what genetic differences are between varieties to start understanding traits and as I explain later, also some aspects of domestication of grapes. One other thing we did over the years was to establish a central repository for all this genomic information. Genomics is it may be very interesting for few in terms of the discoveries you make when you look at genomes. However, the genetic information you develop is useful for many. So it's critical as a genomicist to release this information in a timely way and also in an accessible way. And that's what we created is a website, a web portal called grapegenomics.com, is free to everybody, where all this genomic information is available. And this program has tackled several genomes. Each one of them is part of multiple projects or single projects. So over the years, we gathered funding and support from both the federal government, the industry in California, as well industry abroad. And I will acknowledge the different partners later in my presentation. This is just an example. So every genome, every variety has its own page where you can find information about the articles that presented a particular genome and useful tools if you are into genetics. I know the general audience would not appreciate the value of this, but if you are a geneticist, this is very important, and also the easy access is as critical as the availability. So this is a picture of what we've been working on in the past year. So it's mostly wine grapes. wine grapes, Cabernet Sauvignon was our poster child. We've been investing a lot in the quality of this genome and as well of its relatives, and other wine grapes, and as well as wild grapes. And we're going to explain a little bit why wild grapes are such an important source of information for what we do. All these genomes are publicly available, and the publications are also open source. So if you want to gain more information, you can find the details in the papers listed here. So one thing we started appreciating early on when we started looking at this genome was the extreme genetic diversity. That was actually quite surprising, knowing other crops than grapes. And I like to show this, even if it's not from me, but was from one journalist that actually helped us marketing one of our papers. They came up with this title, "Raising a Glass to Grapes: Surprising Genetic Diversity." Well, that's a good summary of what we started observing when we compare genomes. And forget about all the numbers. It's too much information here. But remember I mentioned that there are major structural variants between genomes that result in changes in genetic information. Well, we started studying these major changes, and we observed actually a striking genetic diversity. And if I would like you to appreciate this number here, about 20% of the genome of Zinfandel is structurally different from the genome of Cabernet Sauvignon. Okay, so who cares? No, well, if you look down in those regions where we have the structural variation, we find genes that are present in Cabernet Sauvignon, but they're not present in Zinfandel, and likewise, and vice versa. So when we just take Zinfandel and we compare to Cabernet Sauvignon, we find the 500 genes of Zinfandel are not present in Cabernet Sauvignon. This does not mean that those 500 genes are actually responsible for all the flavor and aroma differences between the two cultivars. However, it's likely that some of the differences are caused by this difference in gene content between the two varieties. And we've done this exercise across all our collection. And what we started building up is a sense that the more you sequence, the more gene you identify. The more you identify genes that are shared by few cultivars, not by all. And the number of genes that actually shared by all the cultivars decline. And that's what I'm showing here. If you just compare 9 cultivars, you see a decline of number of genes that are shared by all when you compare, for example, the number of total genes that you are start acquiring by sequencing more genomes. And this is, it may sound kind of distant for what we're thinking about, but by doing so, we'll start looking at genes that are associated with phenotypic trait. We can start looking at genes that are only found, for example, in the Cabernet family. And we know, there are characteristic features of that family that are not found in other cultivars. And that's exactly what we're going after. And those important features that differentiate cultivars. What is the genetic background that causes those differences? And you may have differences in gene content. In some cases, we find big changes in DNA structure. This is, again, a simple example where I'm showing that the genetic locus that is responsible for the coloration of the grapes is, and this is millions of letters, is inverted when you compare Cabernet Sauvignon and Chardonnay. Does it have a functional implication on the quality of the grapes? We don't know yet, but by using these tools, we can now appreciate those genetic differences. And interestingly enough, we find this type of inversion always associated at least with the subset of white grapes we have sequenced. So is this inversion important for the quality of white grapes, or the differences between red and white grapes? Working on that. So, and likewise, this is about color, but we know that there are strong genetic differences between cultivars that result, for example, in accumulation of different aroma compounds. So you know that Rotondon, for example, that confers this black pepper aroma to Syrah is pretty much found in Syrah and not in other cultivars. Methoxypyrazines, whether you like them or not, they are typically associated with the relatives of Cabernet Sauvignon. So what are the genes responsible for this accumulation? Likewise for terpenes and aroma in aromatic varieties, etc. So again, this is, for me, is a beautiful exercise because I'm a biologist. I want to understand how the chemistry of the grapes is determined by the genes in the genome. But from an industry perspective, and I imagine that Marianna will talk about that, understanding the genes responsible for the differences in flavor and aroma between varieties provide the information to be able to generate new cultivars by traditional breeding, where those flavors and aromas are combined in unique new fashion. So this is aroma and flavor. Another important trait that we're looking at is disease resistance, which is also very strongly determined by the genetics of the grape and their genomes. As you know, grapes suffer of multiple very severe diseases, and so we basically rely heavily on fungicide and pesticides to control them. The idea of my program and others in the world is to be able to use genetic information to identify other grapevines, wild or cultivated, to introduce in our cultivated germplasm genes that confer resistance to these diseases. So the vine is naturally resistant, and we don't need to protect it with fungicide and pesticides. And we're doing this in focusing particularly on powdery mildew and Pierce's disease. These are both projects are federally funded by the USDA and NSF. Just a picture of where these genomes come from. They come from North America, they come from Central Asia, and they're all from wild grapes, so not in the same species as what we cultivate for wine and table grape production. And likewise, we have this project with NSF, supported by NSF, where we're looking at the natural resistance to Pierce disease in the populations of wild grapes in the southern part of the United States. And again, here the exercise is sequencing genomes, comparing them, and understanding what differences are associated with the resistance or the susceptibility to the disease. And finally, breed new varieties using this information. I would like to conclude with one important part of what we do in the lab, which is understanding domestication. Domestication is the process of selecting and altering the genetic makeup of wild species so they become more apted to cultivation. This is not something that happens in 1 day or 2 days. This is something that happened in thousands of years. Okay, this is due to the fact that the early viticulturists, the early grape gatherers were selecting individuals in natural population that had favorable traits. And by selecting those individuals, now they created a subpopulation that was actually more similar to what we cultivate now than to the wild population where they come from. So again, this is an intellectual exercise. It's fascinating to be able to reconstruct those events thousands of years after. However, it's also an exercise that allows us to understand what characteristics are specific to the cultivated grapes. And this is important because when you compare cultivated versus wild, cultivated grapes are larger in size, are more vigorous. They have, very importantly, flowers with both male and female parts, and I'm going to focus on that in a second. They have way larger clusters with more berries and larger seeds, which probably correlates also with the size of the cluster. So pretty much this looks like something you would buy and cultivate, and this doesn't look like something you would cultivate. Okay, and that means that over time, the early viticulture is selected for individuals that display these characteristics. And domestication of grapes is fascinating. We've been interacting with grapes for very long, probably because we understood early on that grapes were fermenting easily, making alcohol for us. But we think that domestication occurred about 20,000 years ago as a long process. There was not actually cultivation at that point, but was just management of grapes. When I say management, I mean collecting cuttings, going into the next camp, maybe planting, but then moving on to the next camp because people were still nomadic, until we think 8,000 years ago, with the advent of cultivation and stationary life, we had the domestication bottleneck that basically created what we call the Vitis vinifera, this type of grape. So what we've been doing, we've been looking at genetic differences between cultivated and wild grapes, and we started looking at regions in the genome that are actually distinctively different between the two groups. And when we start doing that, we find regions that are actually associated with the sweet one gene, for example, is important for sugar accumulation in the berries. And well, berries accumulate massive amount of sugars, as we all know. And not too much in detail, but also with genes associated with the softening of the berry and other important traits of the berry. So this is just as an example how we can compare genome to understand what may have happened thousands of years ago when the wild grapes were selected. This is a recent work we published where we actually looked at the genetic mechanisms responsible for probably the most important trait that was selected during domestication, which is these type of flowers. So if you look at wild population, the male and the female are separated. Cultivated grapes have flowers with both male and female parts. This is very important because it increases fertilization of the female without the need of having males in your vineyard, as well as bring together important traits associated with the male and the female grapes, which is typically males make a lot of pollen, a lot of flowers, large clusters, female berries, but few of them in small clusters. You combine now these two traits, you have flowers with both female and male parts, but also large clusters filled with berries, which is what we know, what we need. And we did a genomic study. We compare wild and cultivated species, and we identified the genes that are associated with the male sterility in the female flowers and female sterility in the male flowers. They were both lost during domestication. And this is just a curiosity. One of the genes, for example, that make the female male sterile, so make the female incapable of making fertile pollen, we identified this gene that is associated with the permeability of the pollen and the germinability of the pollen during fertilization. Just as an example, how we can use genomic to identify genes responsible for important traits, and importantly for the industry and the breeding programs around the world, when you do this exercise, we also identified the specific gene responsible for it. So now we have genetic markers to select for female or hermaphroditic plants in breeding programs, which was a major limitation before. Most of what I described here is also described in this book that Professor Walker and myself edited. We wrote a couple of chapters, but most of the chapters come from others in the community, and I think it's a good summary of what we know nowadays about grape genetics. I don't have any financial interest in presenting this. I'm proud of it, but I don't make money out of it. Finally, my acknowledgements. I obviously I don't do much in the lab anymore. I wish I still do something, but I'm busy in talking and teaching and doing other things. So all the things I presented today are product of a fantastic and talented group of young scientists that include PhD students, includes postdocs and senior scientists. And for the sake of this presentation, I would like to thank Andrea Minio and Melanie Massonet and Rosa for their important and critical contribution to what we have achieved. Collaborations are also critical, and I would like to mention my two dear collaborators, Brendan Gout from UC Irvine and Andy Walker from UCD, and others in both institutions and others. And finally, the funding. You know, we couldn't do anything without the funding. And again, we've been lucky to have very generous contributions from public agencies, both at the federal and the state level, as well as very generous contributions from the industry. The Cabernet Sauvignon would not be there without the contribution of J-Lore and Gallo. The Semillon genome will not be there without the contribution of the Dolce Winery. And we did a lot also with the international partners, including private companies like Concha y Toro and Viña San Pedro. And that's it, I think. So, Andy, thank you very much for your attention.
Thanks, Dario. Well, thank you very much for your insight into how studying grape genetics can advance viticulture and winemaking. I don't see any questions posted yet, so please post your questions to the Q&A tool. But now we're going to move on to our next speaker, Marianna Fasoli. Marianna was raised on her family vineyard and winery near Verona, Italy. and studied plant science and molecular biology at the University of Verona. She earned her PhD in applied biotechnology while working in the laboratory of Mario Pizzotti. Sorry, Mario. Marianna joined E&J Gallo as a postdoctoral fellow in 2012. Her research focuses on the molecular regulation of grape development and ripening, including the impact of environment, crop load, and cultural practices on the biochemical pathways controlling berry quality. She has also examined key metabolic pathways in yeast that influence wine quality, including molecular recognition of aroma pathways during fermentation. Marianna, the floor is yours.
Thank you, Andy. Thank you for the introduction. So let me share my screen. All right. So today I would like to share with you some of the research that we've been doing at the E&J Gallo Winery to study the molecular basis of grape quality. I am part of wine growing research at Department of Gallo R&D. Micro and systems biology is a functional group of that department, and the research focus is divided basically in two platforms. One is microbiology and biotransformation. For that platform, we have projects related to the optimization of fermentation and processes to improve the quality of our wine and generate product differentiation. And the other platform is systems biology. Systems biology basically is the study of the interactions between the components of biological systems and how these interactions can give rise to the function and behavior of that specific system. Systems biology in wine growing is the study of grape and yeast and their interactions to generate wine. Systems biology studies the molecular networks of yeast and grapes and the influence on quality and the wine style. Basically, we can imagine that there are pathways in a grape berry that can generate metabolite Y and Z from metabolite X with the action of enzyme 1. These metabolites can either be found directly in the wine, like I'm showing here in this example for metabolite Y, or used as precursors by yeast during fermentation. So in yeast metabolic pathways that are active during fermentation, metabolite Z can be converted to metabolite A and B through the consecutive action of enzymes 2 and 3. Metabolite B, together with Y, can then contribute to the complexity of all those attributes like color, aroma, and mouthfeel that we find in the wine. But today, I will focus on the study of the metabolic networks in the fruit. In particular, the goal of our research at Gallo is to understand the molecular and biochemical regulation of key fruit quality pathways for optimizing cultural practices or developing new practices or treatments which can optimize the outcome of those pathways. And finally, also developing new markers to assist our breeding program. We use the approach of gene expression to study the grape quality pathways. One of our foundational learning projects focuses on using gene expression to track and monitor key quality constituents during berry development and ripening, as you can see here. To understand the molecular mechanisms regulating grape berry development and ripening, we created a transcriptomic map. Basically, this means that we mapped the expression of all the grapevine genes, all the genes in the grapevine genome that are active during the development of the fruit. So basically, we performed a very detailed sampling of Cabernet Sauvignon and Pinot Noir grapes from fruit set to maturity across 3 vintages, 2012, 13, and 14. And this was a very detailed sampling, as I said. It was a weekly sampling, so that yielded to a lot of samples, 200 samples that were all analyzed by RNA sequencing. And this study was published 2 years ago in Plant Physiology. So part of this study was the discovery of global and varietal transcriptomic changes over ripening. So I'm going to explain a little later in the next slides what I mean. But first, I'm showing here that we were able to identify 3 main waves of gene expression throughout berry development and ripening. In this plot, basically, the x-axis explains the time in days after veraison, with veraison being that time when the berry starts to ripen and soften and accumulates sugar. And for us, this is time 0, you can see here in the plot. The y-axis represents the extent of the expression, or basically the activity of one gene or a class of genes, a group of genes. To go a little more into the details of what we found, we can see here that the first wave of gene expression was specific to early developmental stages of the berry development. These genes characterize the metabolism of the berry when it's forming and growing, and at that stage accumulates organic acids rather than sugar. The class of genes with this trend can be considered global transcriptomic changes, because we called it global because it's in common between Cabernet Sauvignon and Pinot Noir, with Cabernet being the blue line and Pinot Noir the red line in the plots. The second wave described gene expression trends peaking around veraison or right after veraison. This is a sort of turning point. So veraison is when really the fruit enters the maturation phase and hence starts softening, accumulating sugar, color, and flavors. And this will make the fruit itself attractive for seed dispersion. Some of the genes that were identified during this wave of gene expression were varietal specific. And you can see here in these little plots, red for Pinot Noir and blue for Cabernet Sauvignon. And the trend you see is very similar. And this means that the most of what characterizes the differences between these 2 varieties starts around this phase. In addition to this, though, we were also able to identify some behaviors that were in common between the 2 varieties, as you see in this last plot. The third wave is associated to genes with upward profile over berry development, regardless the variety. The expression trends in the plot are indeed fairly overlapping between Cabernet Sauvignon and Pinot Noir. The functions of these genes are related to the completion of the ripening program, of the maturation program. So just to recap, we were able to identify both the core set of genes of berry development in all the 3 waves of expression, and the genotype-dependent transcriptomic traits were only found at veraison or early maturation phase, namely that second wave. The other project that I'm sharing with you today is the use of gene expression to understand the impact of environment, vine status, and cultural practices on the biochemical pathways controlling berry quality. So I'll show you some results of this study that has a closer connection with the application of these molecular tools. In particular, in the next slides, I'll be focusing on the effect of the environment in terms of intra-vineyard variation. So blocks, like vineyard blocks, are generally not uniform. And a feature that affects the variation in the quality of the grapes is the variability in the vine vigor. This slide shows an NDVI map of a vineyard block. And DVI is an index that measures and estimates vigor of vegetation, so the vigor of the vines. And it's used also to measure the vegetation in the forest, in general in plants, and also in a vineyard block, like in this case. So high NDVI areas that we can see in the map, that are the blue and green areas, are the most vigorous. And in these areas, the vines have the bigger canopies. Low NDVI, and you can see are these red spots in the vineyard, corresponds to positions in the vineyards where the vines have smaller canopies. In particular, this is a Cabernet Sauvignon block, and we focus on 14 specific spots to account for vine vigor and quality variability. To understand quality variability by location in the vineyard, we performed a detailed targeted and untargeted gene expression analysis over berry development at each of those specific 14 spots. We sampled every 10 days from pea size to full veraison, and then at 18 bricks. 18 bricks is a measurement of sugar accumulation, and this corresponds basically to like meat maturation. And then finally, we sampled at 24.5 bricks. That is our sugar target for commercial harvest. An important aspect of the quality of Cabernet Sauvignon grapes is the accumulation of color in the berry skins. You see here how the variability in the field, and these are the spots, influence the accumulation of color by spot. You see that the total anthocyanins quantity ranged from 1.2 to just over 2 milligrams per gram of berry. So like Dario explained it in his presentation, MIB1 is the gene that regulates the accumulation of anthocyanins in the skin of the fruit in red varieties. And it starts doing that around veraison. When this gene is non-functional, that leads to the lack of accumulation of color that we see in the white varieties. In this busy slide, you can see on the right the quantification of anthocyanins at harvest by vineyard spot, and on the left, the trend of expression of the gene MIB1 over berry development by spot. And this is represented by all these lines at different colors. So the overall profile of the gene is maintained. So MIB1 is basically non-active during early development of the grapes. The fruit during this phase is green and hard. But the expression increases sharply as we approach veraison to a peak, and then it either declines slightly or stays at the same level until harvest. We can notice though that the extent of the expression varied by spot. In particular, MIB1 peaked higher for those spots showing the highest color, and the expression was lower for those with the least color. And it is also important here to stress that these changes can be detected about 40 days before harvest. So that concept means that there is a possibility of using gene expression to predict grape quality. If we know the quality marker, we can then track them early during the development of the maturation of the fruit, and possibly use this information to predict the quality of the fruit itself. If this prediction is early enough, we might be able to apply practices or treatments that can modulate and optimize the quality that we find at harvest. Using this gene expression approach that I just showed you, we learned that there are gene expression waves throughout berry development, and this can be global or specific for the variety. This basic knowledge is very important to pave the way to the understanding of the molecular basis of the ripening program, and also all those nuances that are able to generate the varietal differences. We also saw that we can measure gene expression response to intra-vineyard variation, and that can be a tool for predicting harvest fruit quality, and we can do that early in the season. This is instead an example of molecular viticulture, and it shows how we are working to use very advanced tools to complement or even work in synergy with plant physiology measurements and precision viticulture. So, but in relation to Dario's topic, what do we need more and better grapevine genomes for? Well, having reliable reference genomes is important for transcriptomics and gene expression studies in general. The genomic information that Dario and his team is able to generate provides name, location, structure of the genes that we want to track the expression of. And using varietal specific genomes is crucial to understand typicity and varietal traits and their regulation. As you can see here in my example, it is very important for finding and comparing the genes that are specific of Pinot Noir or Cabernet Sauvignon. Moreover, available genomes allow deepening the understanding of quality pathways and the effect of environment and cultural practices. And finally, I did not show an example of this, but genomic studies and the sequencing of new genomes facilitate the discovery of new quality marker genes to be employed in marker-assisted breeding. So basically with this, I conclude my talk, and I really want to thank all the people that contributed to these projects. The entire wine growing research department at Gallo helped generating the results and the data that I shared with you, and much more than what I shared. Really, like, from all the interns that were very, very helpful in sampling in the field during the summer, to the scientists that are, helping me every day with the interpretation of the data. Really thank everybody, and in particular to Bees, Jim and Nick. And last but not the least, also my former colleagues at the University of Verona, Giamba Tornielli, Sara Zenoni, and Mario Pezzotti. And I really thank you all for your attention.
Thank you, Marianna. That was really interesting, and I think it answers our very first question, which was how the information Dario presented could be used to make better wine. But I do want to press Dario just a bit with this question.
You like that?
Huh? Obviously, Marianna has some really, let's say, high-tech tools at her disposal. And if we have this information, this genetic information about grapevines, is it really practical? Will this information lead to something of practical utility to say a more typical winemaker that might be have a family operation as opposed to a large research team that could, is publishing papers. So I'm just wondering how this information may work its way down to the less technically advanced winemaker. Do you have any thoughts on that?
Yeah, I mean, you're right. What Marianna presented is really the top of the line of what you can do. However, I mean, going back to COVID, there are super accurate tests and there are fast tests, and same for gene expression. You can have, what Marianna presented, a global transcriptomic approach, but there are ways to measure individual genes with a targeted approach that I'm not saying that every winery will be able to afford what technology we need right now to do that, but I can see the development of fast markers, whether they are in the form of like a pregnancy test type of markers that allow you to quickly look at the presence or absence or the abundance of a specific gene or its associated protein. And that's a question, I would like, to ask Marianna also if the company itself is thinking about adopting this type of approach to the daily viticulture. That's the way I would like to think about molecular viticulture one day, that all we have done and all the information we have created is actually helping to make intelligent decision on a daily basis. And I don't think we are so far from that. I cannot tell you this is going to happen tomorrow, but technology will help if there is interest. Things move faster then, and I hope this event will create some interest in that direction.
Do you have any thoughts on that, Marianna?
Yeah, it's really the synergy between the advancement of the technologies and also the information. So we really need to understand with these studies that that Dario is doing and we are doing at Gallo, which are these genes that we need to track. And then with the advancement of the technology, I think at some point in the near future, we're going to be able to use these tools and really employ molecular viticulture in the field. And I really hope that at some point, everybody can use these tools. Maybe, not every winery can have a lab where they can run these tests, but maybe there can be like a cheap way to outsource to an external lab, like many wineries do with even wine chemical analysis.
Dario, I've got a interesting question here that's I have to. Is there research into study of traits or genes with other species, related genera inside the same family? For example, I've never heard of this, but maybe you have Cissus genus from Chile. Can we tap these other related vines to gain insight or perhaps capture genetic genes, I suppose?
That's a very, very good question. And yes, the answer is we're doing that right now. We are expanding beyond the varieties, the grape genus, to understand some of the processes that actually help shaping the grapevines. That if you stay in the varieties genus, grapevines, there are different species, but they're interfertile. They look pretty much alike. If you start moving outside of the varieties genus, you start seeing plenty of genetic diversity. They are genetically related, but they are different, very different. Some are desert succulent plants. Maybe flower morphology is somehow conserved, but the overall anatomy and physiology is very different. Mm-hmm. I don't know how to translate that to day-to-day winemaking and viticulture, but for us interested in how evolution shaped the grape genome extending beyond varieties is fundamental, and we're doing that. Still looking for funding, but we started doing it. Yeah.
Okay, I think this one's for you, Marianna. You had these expression patterns of Cabernet and Pinot during different stages of maturation. And this is an interesting question. Do you expect these expression patterns to carry over to other varieties? Because you only explained 2 of them.
Some, yes, there are for sure. And Dario mentioned that too. There are some core genes that describes and allow the maturation of the fruit in general, and that happens in all the grapevine varieties. But then, you know, the more varieties we'll keep studying, the more we'll see those specific traits, those specific genes. And so that core of genes, we'll get more into the details. So those were just 2 varieties. So I hope that at some point we'll be able to have a lot of studies for all the grapevine genomes that Dario is producing. So yeah, and even more.
Okay.
We're not done. We're doing more.
Oh, yeah,
so
sure.
Yeah.
It's hard to keep up.
Good.
Okay, Dario, I have a question for you that I hope you don't get too hot under the collar.
I saw.
But are there any concerns in the GMO community? Actually, I suppose that's the anti-GMO community that you've heard when wine grapes are crossed. And this person actually loved your talk, especially information on Zinfandel, which is his favorite variety.
Yeah. So what I mentioned has nothing to do with GMOs. Not because I'm against or in favor. What we're discussing was gaining more genetic information on cultivars to be able to select progenies from traditional crosses. And traditional crosses does not require any fancy technology. You take the pollen from one plant and put it on the pistil of another plant, nothing else. But when you make the cross, then you want to be able to select quickly the progenies that are better for winemaking, for tolerance to disease and stress. And to do so, use genetic markers. And this is, again, nothing to do with GMOs, is a quick way to select individuals that carry the best genes you wanna select. And I think our research is helping, facilitating that process. Concerning GMOs, yes, what I mentioned is traditional breeding, but if you envision a world where GMOs are allowed and seen favorably from the public, GMOs will basically accelerate what I just mentioned. We will take genes instead of taking the pollen. We will take genes from our resistant variety that is a wild species and put it in Cabernet Sauvignon, and they will be more precise and fast than traditional breeding. But traditional breeding can deliver what we need also. So it's just slower and less precise in the short term. And I hope I answered the question. But,
yeah, so there's a question here from someone whose name looks very familiar to me. They ask, what are some interesting grapevine parents that you'd like to cross for producing segregation populations in future studies?
Is Jerry asking the question? The beauty of grapes, they're all, and it's a technical term, but they're all heterozygous. They all derive from crosses. They are first generation of another cross between 2 varieties. So like us, they have genes from the mother and the father. When you make a kid, the kid will have your genes, the genes of the mother, but also the genes of the grandparents and so forth. So, and they're all connected grapevines. If you follow the phylogenetic tree of grapevines, they're all connected. So I would say there is no bad cross to make, because any cross you make creates diversity in a surprising way. Because, for example, if you take, like in my lab, we took, with Andy Walker, we took Cabernet Sauvignon and Riesling, we crossed them. Well, some look like Cabernet Sauvignon, some look like in between Riesling and Cabernet Sauvignon. Many look like nothing you've seen before. So I think it's key to create the genetic diversity. And then you can use your imagination, what would be the best combination. But really, I mean, in this case for Riesling, for us was to look at terpenes metabolism and aroma. But I would say there is no bad way to do it. I mean, yeah. I don't know if you'd answer, but that's my perspective.
Okay, so I have a question here from Marianna. Were you surprised at the large variation in gene expression across the various locations in the vineyard block experiment? Would you expect more or less variability if the same variety was grown in different environments?
I would expect a very similar trend. There's like vineyard, like environment. So we only studied a very specific part of what the environment can do, and we did see gene expression changes by location in the vineyard. But there are many studies that were done by the Grapevine community in terms of the expression of the genes. If you cultivate the same variety in different locations, you do see changes in the gene expression. So the same genes will behave in different ways. So it's the concept of the genotype by environment, and different varieties will behave differently. So, so it's differences by location in the vineyard, but also if it was two different vineyards, I would find many differences.
I suppose that helps explain why specific environments are ideal for specific varieties.
Yeah,
You know?
even like here in California, right? Like it's very different if we cultivate a variety in the Central Coast or in the North Coast or in the Central Coast. Of course, there's that effect of the weather, that's environment, and that affects the ripening program. And so we do see that in the genes and in the chemistry and in everything that's related to the maturation of the fruit.
We're getting some pretty technical questions here. Marianna, what is the main trigger for what that gene, what did you call it? MYBA1 changes in the field. I think that's the anthocyanin gene. Do you know what the main trigger is? Can you control its expression?
That's a good question. It's hard to say. So MYB1 is actually a controller itself. It's a so-called transcriptional factor. So it controls a cascade of other events. And the most important one is color development. But upstream MYB1, there are other genes that are still unknown. So we have an idea of which can be the genes that trigger the expression of MYB1 when it's time for the berry to mature, but we don't know which are these genes. We know that sugar has an effect, and probably the weather, but in terms of what's happening at molecular level, there are other transcription factors that are probably in charge of turning on the expression of MYB1.
Yeah. Okay, so we're wrapping up here, but I do have actually two questions. I think one of these covers both of the questions. So this is another technically sophisticated questioner. It says, I work in the boundary and science translational space around helping growers adapt to climate change and understand generally about the importance of genetic selection for adaptive traits. But can either of you speak in general terms to how their research can be used to help viticulturalists adapt to future climate conditions, especially conditions we may not even really be able to anticipate?
I can answer, Marianna. I don't know if you want to go ahead.
I can go first.
Okay.
So I think that some of the research that was presented today can generate the foundation of the understanding of the ripening process and of the vines and how they react to the environment. And so once we have a better understanding of those markers, those genes that are involved, that is very helpful when the time of making like new crosses comes. Like we need to find vines that adapt better to climate change or to one of these unknown changes that might happen. If we know better what happens at molecular level, then we know what to select. So this can be my answer. I don't know what Dario can add.
Dario, you get the last word in here.
I completely agree. And I think what we discussed today is really two sides of the same solution, which is one is optimizing viticulture, and I think molecular viticulture should be considered part of the future of viticulture. And on the other hand, selecting plants that are better adapted to the future environment is critical. And I'm not only talking about new crosses and new varieties, but also understanding better the existing varieties, which ones are more adapted. And there are probably thousands, 5,000 or more cultivated grapes all over the world, and in California we cultivate 7 of them. Could it be that we find actually better adapted variety already existing? And I think that will require more testing, more genetic work, and eventually once we have those varieties, molecular viticulture will help very much optimizing viticulture in the field. The thing to keep in mind is everything we do takes time. So the sooner we start and the sooner we think we have a problem and we want a solution, the more likely we survive and thrive.
Yeah, this season has really been, this harvest has really been a difficult one and has reminded us how close climate change is knocking on the door. So I think you're right, Dario. We can't just wait around forever. We really have to get busy understanding this stuff more, understanding this better. So I'm going to close here and thank you guys so much for presenting so much interesting stuff today. Pretty technical, but I think you did a great job of helping our non-geneticists understand how these tools are useful and how they can be used in understanding the grapes. And maybe the next step, we didn't really get into this, was looking at the impact on wine. So thank you very much. And I'll move on now and close the program. So thank you.
Thank you, Andy. Thank you, everyone.
Thank you.
Bye-bye. In closing, I'll mention a few things. Next week, on October 14th, we have our next Sips and Bites with, we'll talk with Napa Valley winemakers and UC Davis alumni, Jill and Steve Mathiasen. Then on November 10, we'll have our fall Savor lecture on how climate change is affecting viticulture in California. So getting back to that climate change question and how UC Davis and the wine industry are working together to address those challenges. About a week after that, on Wednesday, November 18th, we'll have another Sips and Bites, this one on distilled spirits, getting ready for holidays. And we'll be tasting pear eau de vie cocktails. Please check your email messages for reminders about these wonderful upcoming events. And of course, if you're on Facebook, you can follow us and you'll get reminders there as well. On your way out, please fill out our brief survey to help us guide future programs. Thank you very much for attending, and good night.